And today I would like to tell you about our recent progress on the Auxin-Inducible Degron project. So let's get started. And also, if you have any questions when I'm giving a talk, you can just interrupt at any time. Please speak to me by switching on the mic and the camera, please. Right, so let's get started. Okay, so here you can see a watch. So that is a mechanical watch, and if you see behind, you can see small bits and pieces. And if you want to understand the function of, for example, this rotating wheel, so there are two ways you can do. So one is observation, and the other one is perturbation. And from the observation, you might come up with a prediction or model or whatever. But to prove it, you have to perturb the system to see what happened, okay? And this is exactly the same when you want to study the function of a protein of interest in living cells. And I've been working on DNA replication for the last 20 years since I was a postdoc. And proteins involved in DNA replications are, in many cases, they are essential for cell viability. Therefore, you cannot make gene knockout. So in that case, what you can do is to use a conditional system. And there are many conditional systems existing. The most popular one is like an siRNA. And if you are working with mice, you might be using CLE-mediated conditional knockout systems. But all these systems works either at DNA or messenger RNA levels. Therefore, even if you shut off the transcription, for example, the protein still exists, and then it gets reduced depending on its half-time, and sometimes it can be quite slow. And sometimes this can be quite problematic because with reduced amount of the protein, cells might accumulate some problems, and you might be seeing a secondary defect. So to see the direct consequence, it is important to deplete a target protein in a really short period of time. To achieve this, now the protein knockdown systems are drawing a lot of attention. And the Degron, such as Auxin-Degron system, AID, DTAG, and DD are such systems. And today I would like to mainly talk about this AID system. And then alternatively, the PROTAC, it's a chemical biology system. PROTAC is also drawing a lot of attention, in particular in the pharmaceutical field. And what is Degron? So Degron is a protein domain or tag that can induce degradation, okay? And the great point about the Degron is it works in a really rapidly and sharply. And then also, it can work in a reversible manner. And then also the fusion protein, the expression level of fusion protein can be tuned, okay? The first generation of the Degron system is not AID system, actually, unlike Kaz told you. The first generation of the Degron system was originally made by Alex Walshawski in early 90s. That is called temperature sensitive Degron system, or TS Degron. So this system works very well in body cells, and I can explain a bit. So this TS Degron system, so you can place a Degron cassette, TS Degron cassette at any terminus coding region of the target gene. And this TS Degron cassette is composed of two parts. The one is this ubiquitin gene, and the other one is a temperature-sensitive version of DHF4. And the fusion protein will be transcribed, and then this ubiquitin will be cleaved by end processing mechanism, exporting arginine residue at N-terminus. And this arginine will be recognized by an easily ubiquitin ligase called UBC1, UBC2. However, at lower temperature, like at 24 degrees, the fusion protein is, in many cases, is stable and functional. But once you raise the temperature to high temperatures, such as 37 degrees, a conformational change in DHFR is induced, and exposing lysine residue outside for rapid ubiquitination and the destruction. So here you can see a fusion protein here that is stable, but once you raise the temperature within 30 minutes, this fusion protein is gone. So I can show you one example how powerful this Degron system was. So we decided to target this replication factor called MCM227 complex, which is a key component, a key master factor of DNA replication. We decided to make a 2 mutant of one of the subunits called MCM4. So this is MCM4 temperature sensitive Degron mutant at the higher temperatures. So these mutants cannot form colonies at all. We also generated a mutant so by replacing the promoter with tetracycline inducible promoter. So in the presence of doxycycline, again, this mutant cell lines do not form colony at all. And if you look at the protein, so this is a fusion protein of the TS Degron protein. So the expression can be seen at 0 time, but once you raise the temperature within 2 hours, the fusion protein is completely gone. On the other hand, in case of tetracycline inducible promoter, by adding doxycycline, so this is the MCM4 protein, this lower band, even at 4 hours, still you can see the bit of expression of MCM4. This makes a difference in the phenotype. In case of temperature sensitive Degron, so this is a FACS profile of body cells, and the first peak correspond to G1 phase, and second peak correspond to G2M phase and S phase in the middle. And once you raise the temperature, you can see cells are arrested at the boundary between G1 and S phase, showing that they have got a strong replication defect, okay? On the other hand, when we shut off transcription of MCM4 by adding doxycycline, cells are arrested at G2M phase. This is because with reduced amount of MCM4, cells can still carry out DNA replication, but they get some problem during the S phase. And then this makes a kind of DNA damage, and then checkpoint signal was activated. As a result, cells are arrested at G2M phase. And there is an important message. So even if you target the same protein, if you use a different techniques, you might see a different result, okay? And if you want to see the direct consequence of the depletion, it is important to deplete your target protein in a short period of time, okay? So 20 years ago, I was very happy to use this technology to study replication proteins in body yeast, but eventually I started to feel a bit of frustration. I can show you the reason. So the first reason is because we can place this TS Degron cassette only at the N-terminus of the target protein. So it doesn't work at the C-terminus. And then also, the system works very nicely in body yeast cells. However, because temperature shift is involved, we can't apply this system to other eukaryotic systems, such like human cell lines. So I've been wondering for many years and thinking that if we could generate different type of Degron system, that's going to be quite useful for many studies. And in the end, we came up with a new idea to use a degradation system derived from plants. So this is the principle of Auxin-Inducible Degron or AID system. So in plants, there is a very unique degradation pathway existing that is controlled by a plant hormone called auxin. The chemical name is indole acetic acid, or IAA. So this auxin activates an E3-ubiquitin ligase containing this subunit called TIR1. And what happened in plant cell is this AUXIA, that is a transcription inhibitor, that's going to be recognized and ubiquitinated for rapid degradation. And now transcription factor ARF is free for activating downstream genes. But this is the mechanism how auxin works in plants. And we thought that we might be able to transplant this degradation pathway into other eukaryotic systems, including mammalian cells. And what we did was we expressed this TIR1 cloned from rice, and the other components are well conserved in all eukaryotic species. So therefore, we can establish a chimeric easily ubiquitin ligase like this. And we can fuse a Degron tag derived from this AUXIA, and then this fusion protein is stable without auxin. But once we added auxin to culture medium, the easily ubiquitin ligase is activated for recognizing this Degron tag, and then fusion protein going to be ubiquitinated for rapid degradation. So this is the system how AID system works. So now we can generate a human mutant cell line. So what we do is like this. So we can make our cells expressing this ubiquitin ligase TIR1 by introducing this TIR1 expression cassette at a safe harbor locus, such as AAVS1 locus, by using CRISPR-Cas. And this is going to be the parental cell line. And in this parental cell line, we can fuse a Degron tag. This MID tag, the size is about 7 kD. So we can place this tag either at C-terminus coding region or N-terminus coding region by using CRISPR-Cas. And then now you can make this type of mutant. So I can show you one example. So here we decided to target this protein called RED21 cohesin. So cohesin is a ring-like complex, which is important for making sister chromatic cohesion and important for making higher order chromatin structure. So we fused this Degron tag at the C-terminus of RED21 together with a fluorescent tag called clover. And now you can see this RED21 fluorescent signal in nucleus. And by adding auxin, we can achieve really nice rapid depletion of RED21 cohesin. So here we quantify the data, and within 60 minutes, most of the protein is gone. All right. And we published this paper some time ago, and we deposited all materials at Adgene. So now you can access those materials. And so far, I think there are more than 3,000 distribution has been done. And interestingly enough, so the more than 90% requests came from either US or European country. And I'm based in Japan, I'm Japanese, but somehow this technique is not very popular in my country. So I just would like to know the reason why. But anyway, so that's one of the reasons why I'm speaking to you today. And so far, I've been telling you good points of the AID technology. However, but eventually we started to notice that there is a bit of problem with the current AID technology. So I can show you some negative points. So we noted this problem when we made a mutant of dynein-heavy chain 1, DHC1. So that is a motor protein. So again, we decided to fuse the Degron tag and the fluorescent clover tag at the C-terminus coding region of DHC1. And here we use two types of parental cell line constitutively expressing TIR1 under the control of a CMV promoter, or conditionally expressing TIR1 under the control of tetracycline inducible promoter. And after selection, so here you can see plates. So this is a control experiment without CRISPR-Cas, so we shouldn't get any colonies at all, which is good. But when we use the cell line, parental cell line constitutively expressing this TIR1, we don't get colonies here. On the other hand, when we use a cell line conditionally expressing this TIR1, we got quite a lot of colonies. But the reason is because, so actually sometime just by fusing this Degron tag, even without auxin, a bit of degradation happens. And the DHC1 is very important for cell viability. Therefore, cells acquire this, DHC1 Degron fusion protein died here. So we took colonies from here and look at the growth of the cells. So here you can see without any treatment, cells can continuously grow, which is good. And by adding doxycycline for inducing ubiquitin ligase TIR1 together with auxin, cells stop cell cycle after one round of cell cycle and it started to die. So that is good. But if we added just only doxycycline for inducing this ubiquitin ligase, so we started to see partial growth defect. And at this point, if you look at the cells under the microscope at this point, you can see quite a lot of rounded cells. So they are arrested in mitosis. The reason is because DHC1, dynein, is very important for forming mitotic spindle. So therefore, they are arrested. And if you look at expression level by Western blot, so this is DHC1 fusion protein. In this case, we use tetracycline instead of doxycycline, but just adding tetracycline significantly reduce the expression of the fusion protein. So the problem associated with the current AID technology is there is a bit of leaky degradation happening, and even without the addition of auxin. And sometimes you might see some phenotype, and sometimes it could be quite lethal. And then also the concentration of auxin is relatively high. So we typically use like between 100 to 500 micromolar of IAA. And that is not very good for some cell lines. And then also nobody has successfully applied this AID technology to adult mice so far. Okay, so we wanted to improve this system for many years. And eventually we managed to make a new version of the AID system. That is called now AID2, okay? The difference between the original AID system and the AID2 is there are 2 points. The first point is we use now a mutant version of TIR1, so which have got a mutation at the ligand binding site here. And this ligand binding site mutation creates a hole, and therefore now the mutant doesn't work with the original auxin anymore. However, it can be reactive with an analog, auxin analog having a bump at here, okay? And we were originally inspired by a recent publication by a group in Nagoya University. So they made a kind of mutant of TIR1 in Arabidopsis thaliana, and they screened a chemical that can be reactive to this type of mutant. So inspired by this paper, we decided to apply the system to human cell lines. So what we did was we initially established a reporter cell line. So this reporter cell line expresses this Degron tag together with GFP, okay? And we subsequently introduced either wild type or mutant version of TIR1 at the safe harbor AABS1 locus. And we look at the fluorescent signal in this reporter cell line. So here is a result. So here you can see this is the expression level of the reporter on the original reporter cell like this. When we introduce this wild type of TIR1, we can see significant decrease of the expression. So this is the leaky degradation, as I told you before. And by adding 100 micromolar of IAA auxin, we can induce nice degradation like this. So on the other hand, when we introduce 2 types of mutants, F74G or F74A mutant, the expression level is comparable to the original reporter cell line. And now we can induce very nice degradation with quite low concentration, 1 micromolar of one of the ligand called 5-PHIAA. Here you can see. Now we think now F74G mutant is a better option than F74A because F74A mutant is still reactive to auxin for some extent. So we treated this reporter cell line with different doses of the original auxin, and here is the data you can see. So compared to the F74G mutant, F74A mutant shown in green is more reactive to auxin. And this leaky degradation sometimes is induced by auxin-like chemical existing in culture medium. So that's the reason why we prefer to use this F74G mutant in the following experiment. And also we wanted to identify the best ligand for inducing degradation. So this is the original AID system with the wild type TIR1, and we treated the cells with very low concentration of auxin IAA. So commonly we use this high doses, but with this reduced low concentrations, we don't see much degradation, okay? So in this case, we use this mutant version of TIR1 F74G, and we use 1, 2, 3, 4, 5 different types of ligand, potential ligands, and at 50 nanomolar, so it can induce nice degradation, okay? And we titrated, and we found that this 5-PHIA works the best among the 5. If you look at the data at 1 nanomolar, so the other ligand do not work well, but it's still reactive for some extent. So therefore, we decided to use this 5-PHIA in the following experiment. We think that this 5-PHIA is not toxic to human cell lines, HCT116 cells. So we treated this original HCT116 cells or cells expressing this wild type TIR1 with different doses of original auxin IAA. And at higher doses, we see there is a bit of a defect in colony formation. And in case of 5-PHIA, 1 micromolar is the highest dose we use throughout the experiment, and we don't see any defect in colony formation. We also look at the transcriptome to see the effect on transcription. And when we treated cells with 100 micromolar IAA, a bit of genes are affected, but that was significantly reduced when we treated cells with 1 micromolar 5-PHIA. So we think that 5-PHIA is a safer option compared to the original IAA. And then also with the new system, we can induce a very sharp degradation at the low concentration of inducing ligand 5-PHIA. So the pink shows the new system. So the original expression level of the reporter is much higher compared to the original AID system. This shows the difference of the leaky degradation. And we can induce a very sharp degradation at low concentration of 5-PHIA. The original system works, but this range at the higher concentration of IAA. And if we compare the difference of the DC50 value, we think that 5-PHIA works about 700-fold better than the original system. And the degradation kinetics is faster. So the expression level is higher because of the suppression of leaky degradation with the new system. And we can induce sharper degradation, sharper and faster degradation. In this case, the half-life is about 70, 80 minutes. But please note, the expression level of this reporter is extremely high. That's the reason why the half-life is rather long. So you will see when we target the endogenous proteins, we see much shorter half-life. And then this system works in a reversible manner. We can induce degradation here. And then by washing out the 5-PHIA from culture medium, we can see the recovery of the reporter expression. Almost 100% recovery was seen within 4 to 5 hours. So until now, I've been showing you the data obtained by using human HCT116 cell line, but this system works in other cell lines as well. So I can show you one example when we use mouse hippocampal neurons. So we introduced a cassette reporter protein together with a wild type or mutant version of TIR1, and we looked at the fluorescent signal. So here you can see the data. The original expression level of the mutant is much higher compared to the original AID system. And under this condition, somehow, the original AID system didn't work well. But on the other hand, with a new system, we can induce very nice degradation like this. And we think that F74G mutant works slightly better than the FT74A mutant. Okay, so until now, I've been showing you the data with the reporter protein. And the next question is whether this system works to control the endogenous proteins. So to answer this question, again, we decided to target this RAD21 cohesin protein, okay? And here you can see the expression of RAD21 cohesin under the microscope. And you can appreciate that expression level of RAD21 is higher with the new system. And then degradation can be sharply induced compared to the original AID system. So here we can see from this graph, the original expression, the expression level of the new system is much higher, and we can induce very sharp degradation. The T-half of the new system was about 12 minutes. That is almost about a half of the half-life of the original AID system. So we can induce even faster degradation with a new system. And after degradation, like with a new system, after 1 day, what you can see is almost 100% cells are arrested in mitosis. And if you look at the chromosomes, so here you can see in the control experiment, X-shaped chromosome, because sister chromatids are aligned together by cohesin. However, when we treated cells with 5-PHIA, so now RAD21 cohesin was destroyed, therefore cohesion was completely lost, as you can see here nicely. Okay, and the next we wanted to know whether we can generate a DHC1 mutant. So remember, as I told you before, DHC1 mutant is a little bit difficult to generate a mutant cell line with the previous system. So again, we targeted this DHC1 with Degron and Clover, and we use a parental cell line constitutively expressing the original TIR1 or TIR1 F74G mutant. And here you can see the result. So this is a control experiment without CRISPR, so we don't see any colonies at all. That is good. And then, as I told you before, when we use the cell line constitutively expressing wild type TIR1, we don't get any colonies again at all. But when we use the mutant expressing OSTIR1 F74G, we got the multiple colonies. We took clone and look at the expression of the fusion protein. So this is a wild type DHC1 that was a little bit sifted because we fused the Degron tag. And by treating the cells with 1 micromolar 5-PHIA for 4 hours, the fusion protein disappeared. We confirmed degradation under by flow cytometry as well. And after 1 day, almost 100% cells are arrested in mitosis because mitotic spindle was completely disorganized, because dynein is very important for establishing nice mitotic spindles. Okay, that is good. We furthermore generated more mutants by tagging endogenous proteins. So this is a case of CTCF. CTCF is important for making higher order chromatin structure together with cohesin. So we fused Degron tag to the endogenous CTCF, and the fusion protein disappeared nicely. And so this is SMC2, that is a condensing subunit. Again, the fusion protein disappeared really nicely. And another example is here. This is RNA polymerase 2. Again, the polymerase 2, RNA polymerase 2 largest subunit, sorry. Also, the fusion protein disappeared. So all of these 3 proteins or genes, actually we couldn't generate a nice mutant with the original AID system. But now with the AID2 technology, we can generate a very nice mutant. And in this mutant, we can deplete the target protein in a short period of time. And two more examples. So here we decided to target a transcription mediator called BRD4. The fusion protein of BRD4 disappeared nicely. And another protein is topoisomerase 2. Again, the fusion protein disappeared nicely. And these two proteins are really is a good target for cancer chemotherapy, actually. Therefore, we wondered whether we can model cancer treatment by a xenograft assay. Therefore, we transplanted these mutant cell lines to nude mice. And after 1 week, you can see a tumor was formed like this. And then we started to inject different doses of 5-PHIAA by IP injection every day. And then after another 1 week, we sacrificed the animals and looked at the tumor. So this is a control experiment when we transplanted the parental cell line, okay? So tumor grew even if we treated the highest dose of 5-PHIAA. And this is the tumor weight on the final day. There is no differences among these conditions. On the other hand, when we use this BRD4 mutant cell line at any doses of 5-PHIAA, we can see significant suppression of tumor growth. And when we look at the tumor weight on the final day, you can see there is a significant difference compared to the control experiment. So the same is true in case of topoisomerase 2 mutants. We see significant tumor suppression, and the tumor weight on the final day, you see significant difference in all conditions compared to the control experiment. Now we can model this tumor growth assay by using nude mice, and it also shows this system also works in mice. But we wanted to confirm further whether this system works nicely in mice or not. So to answer this question, we decided to make a transgenic mice. So what we did was we introduced a set containing this TIR1, either wild type or F74G mutant, and then also together with a reporter containing GFP and Degron. And after transgenic injection, we got multiple pups. And when we introduced this wild type TIR1, what we noted was none of them, we didn't get any transgenic positive mice at all. On the other hand, when we introduced this F74G mutants, we got multiple transgenic lines, and many of them are GFP positive. So from this experiment, we noted that when we introduce this wild type of TIR1, some problem might be happening during the embryogenesis, and that might cause lethality. That might be the reason why, until now, nobody has successfully generated the AID mutants of AID mutant mice until now. So we got multiple GFP positive clones. We sacrificed one of them to look at the fluorescent signal in multiple organs. So here you can see the fluorescent signal coming out from multiple organs, and we injected 5-PHIAA by my IP injection, and after 6 hours, so we noted that in multiple organs, the expression level was significantly reduced. So we quantified the data, and in many cases, more than 90% of the signal was gone by injecting 5-PHIAA, except for the brain. In case of brain, we achieved only about 50% of the degradation compared to the original expression level. The reason is because we think that the transfusion of 5-PHIA to the brain was not very efficient. So we looked at the concentration of 5-PHIA in multiple organs, and we noted that the concentration in the brain was relatively low compared to other organs. And also we can really induce rapid degradation even in embryos. So we crossed one of the TG line, and the pregnant mice was treated or injected we injected 5-PHIA to the pregnant mice, and we looked at the fluorescent signal in embryos. So here you can see fluorescent signal that was almost completely erased within 60 minutes. We looked at the GFP signal at a different stage of embryogenesis, and again we can see a nice degradation within 1 hour. And we confirmed this data by Western blotting as well. Here you can see within 1 hour the reported expression was almost completely gone. Okay, so this is a summary of my talk. So with the AID2 system, so the great point about this AID2 system is we don't see leaky degradation almost at all anymore. It works at the very low concentration of new ligand called 5-THIA. Typically, we use about 1 micromolar. And the degradation kinetics is actually faster than the previous system. And I didn't show you the data, but the AID system works nicely in body disease as well. But also, we show it works in mammalian cells, and more importantly, the new system works in mice as well. Okay, and we are very happy to distribute the all kind of material for your studies and to distribution to the academia. So the plasmid can be requested from, you can obtain from either Adogene or RICAN BRC. Those are BioResource Center. And cell lines described in today's talk. So I'm happy to distribute, please write to me, and distribution to the companies. So we can do as well. We're very happy. Please write your wishes to the Intellectual Property Department of our institute, NIG. And if you are interested in collaboration, I'm very happy to have a discussion anytime. So feel free to write to me at any time. So in the end, I would like to acknowledge people involved in this project. So the cell experiment with HCT116 cells were done by ex-student Aisha and the current postdoc Yuichiro. And the experiment using mouse xenograft assay was done by Naomi Kitamoto. And the TG mice experiment with TG mice was done by Yumiko Saga and Reiko Ajima. And transcriptome analysis was helped by Hirofumi Nakaoka. And all chemicals we used in this project was synthesized by my nice, great collaborator, Kenichiro Hayashi. And experiment with mouse neuron line was done by Haruki Takeuchi and Risako Nakano.